One day, on a sunny day, there are 4 friends, which is Dini, Ilya, Mira, and Mija. But then, Dini was craving for papaya.
I'm craving for papaya.
Mija, let's find the papaya for Dini.
Okay, let's go.
It's papaya.
It's delicious.
Dini, do you know that papaya has many benefits?
No, I don't.
In papaya, we have vitamin A and papaya enzyme that make our flawless skin. There is still have the balance of papaya. Do you have any ideas what we can do with this papaya?
Why not we do scrub at Ilya's face?
My face? Sure, why don't? Because I haven't applied scrub on my face for a while. So let's do it.
Sure.
Sure, why not? Do you know, guys, that papaya is one that uses the DNA recombinant technology?
What is that, Mira?
Look up.
The definition of recombinant DNA technology, a method in which DNA molecule made in vitro with segments from different sources or by combining genes from different organisms.
Now, I will explain about DNA recombinant technology. Here, I have been listed the 5 of tools that involve in DNA recombinant technology. So, for the first one is target DNA, second is restriction enzyme, third is DNA cloning vector, fourth is host cell, and the last one is modifying enzyme. And then, this is the methods in DNA recombinant technology. The first one, we will choose the gene of interest. So in our experiment, we have been choose the papaya gene of interest. So this is the first step, which is the new isolation of gene. We will take the gene of interest from the papaya, and then the second one, this is the bacterial cell. We will take the plasmid from the bacterial cell. We will take out the plasmid from the bacterial cell, and then the plasmid will This is the plasmid that we have been taken out from the bacterial cell. And then in the plasmid we have like Z. Then MCS. Okay. So we have these items in the plasmid, which is the Z. MCS is the multiple cloning sites, and then MR is the ampicillin resistance, and then this is the origin of replication initiation. So the function of the multiple cloning site is able to accept the foreign DNA, and then the ampicillin resistant is have selectable genetic marker, and then the origin of replication initiation able to replicate fully in the host cell. And then in the plasmid also have the bases sequence, which is the name is palindromic bases. Palindromic. What is the palindromic? Palindromic is the same bases of DNA strands, but from the opposite direction. For example, this is we call palindromic palindromic, because this is the DNA strands, and then from the opposite direction it also the same basis. So this is the palindromic. Okay. And then target DNA, okay. And then the tools for the second is restriction enzyme. So for the plasmid, we will use the eco RY. So it have two types of cards, which is sticky end and okay. The bacteria that we use for the sticky end is the bacteria that we use for the sticky end is eco RY. So for the blunt end, we use the small white. Okay, these are the types of bacteria, and the types of bacteria will be the restriction enzyme that will cut the restriction site. Okay, for the sticky end, the DNA bases will be GGA, TCC. So CC, TAGG. So the ways that we cut for the sticky end is from here to here. So it will be G C C T A G and G A T C C G. Okay, this is the ways to cut for the sticky end. And then for the blunt end, the DNA is T D G G C C A, then A C C G G T. Okay, for the blunt end, we will cut at the center. So like this. It will be to like this. So for the second step for the DNA recombinant is cut and cleave, which is the plasmid that we take out from the bacteria cell will be like this, because it will be like this because we have been cut the restriction site by using the restriction enzyme. So we have two, which is the sticky end and blunt end. So if we use the sticky end, we will use the EcoR1 as a restriction enzyme to cut the restriction site. And then for the blunt end, we will use the small one as a restriction enzyme to cut the restriction site. And then after we have been cut, it will be the restriction fragment. This is also same. Okay, and then so this is the second step. Cut or cleave. Okay, second. And then the plasmid also will be the vector, then DNA cloning vector. Okay, switch the enzyme. Okay, DNA cloning vector. And then after we have choose the gene of interest, we will take the gene of interest to add into the restriction site of the plasmid. So here it will join by using DNA ligase. So DNA ligase will join them. So it will be like this. This is the plasmid. This will be the gene of interest of papaya, because we use the papaya. Okay. This is we call the third step, which is insertion. Okay, insertion. And then the gene of interest will join with the plasmid by using DNA ligase. Okay, so we have insert the gene of interest of papaya in the plasmid by using the DNA of ligase. The name of this will be recombinant plasmid. So this is the recombinant plasmid. So this is for example, this is the plasmid that we have been cut by using the restriction enzyme. So this is we call the restriction fragment because we have been cut at this side. Okay, so this is the gene interest, and this is the plasmid that we will combine it by using the DNA ligase. This is the example for sticky end because the way that we cut is like this. So this is where the result when we have been joined them by using the DNA ligase. For this example, we will use the stapler as an example for DNA ligase. So when we have joined them, this is we call as a recombinant plasmid. So this is this is the types of sticky end, and we use the EcoR1 bacteria. So this is another example. For the second, we use the types of cuts, which is the blunt end. So we use the small one example, small one bacteria. So we cut at the center. This is the restriction site, and then we cut by using the restriction enzyme. So this is the result by, so it will be the restriction fragment. Okay, it different from the sticky egg. And then this is the gene interest that we will join with this. So this is the result of blunt end cuts. We have been joined by using the restriction enzyme. We use this, in this case, we use the stapler as the enzyme to join them.
So now we're going to talk about the host cell. Okay, this is the host cell. Okay, a host cell is actually a cell which can be replicated and allows the produce of recombinant DNA. Okay, its characteristics is able to receive recombinant DNA through transformation process. It is also able to maintain the structure of recombinant DNA from one generation to another. And also, it is able to amplify the gene product from the recombinant DNA. The reason why bacteria is the most commonly used as a host cell is because that the DNA can be easily isolated from or introduced into a bacterium. Okay. And bacterial cultures grows quickly, rapidly, replicating any foreign genes that they carry. Okay, for example, like E. coli. This is the E. coli, and this is the recombinant DNA. Okay, the host cell is then undergoes transformation and amplification process. Okay, the DNA recombinant is then inserted into the bacteria through transformation process. As the bacteria reproduce, the recombinant DNA is also replicated, or known as clone. Okay, the next step is screening. Okay, in order to identify the bacteria clone containing recombinant DNA, blue-white screening is done. Okay, we will place the bacteria in a plate on an agar containing antibiotic, ampicillin, sugar, and agar. We will incubate until colonies grow. If the bacteria inside the plate on agar is not resistant to the antibiotic ampicillin, the bacteria will not grow. But if it resistant to the antibiotic ampicillin, the bacteria will reproduce and form colonies. Okay, and then if the colonies without recombinant plasmid will be blue, while colonies with recombinant plasmid will be white. Okay, for colonies without recombinant plasmid, it contains intact Z. It is also can produce B-galactosidase and also can hydrolyze X-gal. Okay. For colonies with recombinant plasmid, it is do not contain intact Z. Also cannot produce B-galactosidase and also cannot hydrolyze X-gal. Okay, the DNA recombinant is then joined by the DNA ligase, catalyze the formation of phosphodiester to form recombinant DNA. also from me, Dini,
Mija,
Mira.
Ilya.
Thank you. Bye.